Today I'm joined by two of the minds behind LoopDx, a Barcelona-based startup that is rethinking how we diagnose sepsis by looking at patients' immune response instead of just hunting for pathogens. First, Enrique, is the co-founder and CEO, is an immunologist and a biotech entrepreneur. His research on how the immune system behaves in severe infections led directly to LoopDx SeptiLoop test for ultra sepsis detection. And alongside him today is Eduard, co-founder and chief operating officer, a pharmacist with health innovation specialists, who has helped turn this science into a CE-marked point-of-care diagnostic in that hospitals can actually implement at the bedside. Together, they're building a technology that aims to catch sepsis within hours rather than days, potentially saving countless lives and reducing the huge clinical and economic burden that burdens the healthcare systems of the world. Hello, gentlemen.
Hello.
Hello, thank you for the invitation.
Happy to talk with you.
Okay, pleased to meet you. Eduard, can we start with you? If you wouldn't mind telling us a little bit about yourself and how you ended up where you are today, please.
Sure. So my background, I'm a pharmacist, graduate here in Barcelona. So let's say my pathway to move over innovation comes from an exchange program that I did to UT, University of Texas, where I discovered the transfer kind of mark of from science to private companies and how this affects the university inflows with licenses. And I was interested by that. So then I went into a master program in Barcelona called The Health, which is a biodesign labeled program for healthcare innovation. And over there is where I met with Enrique. That program pushes the different groups of people that are from different kinds of background each other and different, like usually it's engineers, designers, scientists, and business. They make like a small group, like to simulate a future startup, and they push us to find unmet needs. And over there, like Enrique and I, we were both with each one in different projects, but we just have nice chemistry together. So after the program, we decided to join forces. Enrique started the company, and I joined it like a little bit afterwards. Then from there, I've been helping the company as a co-founder, COO in operations, legal department, and human resources.
Perfect. Thank you very much, Eduard.
Sure.
Enrique, could you tell us a little bit about yourself, please?
Yes. So my background is I have a PhD in immunology and infection disease. I have been working in a hospital laboratory research up to 2020. So doing this research in how the immune system responding anti-infection. We have more than 25 publications in the area related with sepsis and immunology. And my motivation was to create something that really applied to the hospital. Is where we moved from Madrid to Barcelona, where we met Eduard in the program, and we will discover this part that from the laboratory is not really understanding. No, what is the unmet need in the clinical practice? And during this program, we discovered that one of the pain points is that the current microbiology tests spend a lot of time to provide the result, and also the methodology is slow. And with this idea and combining the two walls, my scientific wall and this unmet need wall, we we moved to develop a product that is working with the immune system response in order to provide a indirect result of if the patient have blood testing infection. So after this Barcelona accelerator program, we moved to a lot of European program, European grant, and also we start collaboration with a hospital who co-develop, co-owner the patent, our first patent in the company, and this is the initial. So the company was built in the end of 2018. And this is a long process to have from scratch, a product until the market that we are.
Absolutely. That's a very difficult pathway you've been down, but you've been quite immensely successful with some of the publications. Tell me, LoopDx is shifting sepsis diagnosis from pathogens to immune status. Now, in one sentence or a couple of words, can you tell me what problem are you fundamentally solving from the current sepsis tests? What the current sepsis tests miss that yours picks up?
So it's the initial triage screening, where the typical patient with suspected sepsis have some symptoms, but they already have a local infection. So the physician have to understand if this infection is local or is moving or is transforming to systemic. In this point, there are a lot of gray area of patients with not clear symptoms, and this is the problem of the difficult to early identify sepsis, that right now the tools available in the market are more focused to the organ damage. Even the clinical score are focused to organ damage in the body. So this means that the damage is already there, so it's late for early identification of sepsis.
Yeah, so in summary, early identification of the disease at the early stages of the disease.
Perfect. Okay, that's very well said. Thank you. So for a clinician in the emergency department who has never heard of LoopDx, how would you explain what SeptiLoop actually measures and what results they see at the bedside in under, say, 30 seconds or 2 or 3 minutes?
So the job to be done for an emergency physician is to risk assessment and identify the patient that need hospital care or can discharge to home. So SeptiLoop provide immunosuppression, early immunosuppression that is caused by initial early sepsis. And this result is totally novelty for a physician and is to pay attention or monitoring in detail this patient, even recommend new tests like the physician don't initially understand, because SeptiLoop provides the result of this immune deregulation that happen in the early phase of sepsis. Okay,
all right, that's perfect. So you claim detection as early as 1 hour after infection, which is 3 times higher. You have 3 times higher accuracy than current tests. So where does that performance come from scientifically, and what data are you most proud of?
Our main clinical studies was performed in the emergency department from 2 hospitals in Barcelona, Partaulí and Bellvitge, and each study have different final goal standard classification of sepsis. So the performance depend of this final classification of sepsis. For example, in Bellvitge, the final diagnosis was sepsis 3 criteria and positive blood culture in the first 24 hours. So our device will be able to predict this sepsis 3 and blood culture positive from the emergency department with a point of care test with 91% of sensitivity and 80% of specificity. And in case of the Tauli study, depending of the gold standard used, sepsis 3, sepsis 2, the performance will change a little bit, but yes, we will have from 70 to 90% of sensitivity in this case. Even in the patient that have low symptoms level, like for example, NEWS score less than 5, QSOFA less than 2, also the SeptiLoop performance is maintained.
Okay, Eduard, do you want to add anything?
Yes, well, the sensibility that Enrique discussed comes from this clinical study of Hospital of Bellvitge. Here, and the main differences, as Enrique well stated, are the final diagnosis of sepsis and also the recruitment of the patients. In the first one was patient recruited in the emergency, the one from Bellvitge, recruited from emergency department patients that were positive in blood culture, but from the same sample that at early emergency, the first sample that they collected this blood culture. So when it turns to be positive with this sample up to 16 hours. It's the same sample, the one that we used for recruitment from performing SeptiLoop. And in case of the hospital of Partaulí from Sabadell, here, as Enrique well stated, the inclusion criteria was patients that were suspected from sepsis from the emergency department with lower scores of NEWS. In this case, was a score of NEWS 3 or superior.
Okay. So from what I've read and what I know, LoopDx focuses on immunosuppression and lack of TNF-alpha production. Okay, so as a biomarker, why is this the right biosignal or biological signal, I should say, versus other immune markers you could have had chosen?
So yeah, the biological validation of this methodology is dependent of the publication, but it's called endotoxin tolerant status. Is the ability of the cell to block this initial TNF-alpha response, and this will be maintained blocked in the cell, in the innate cell, during the up to 7 days. So with this initial mechanism is happening in a very early phase of sepsis. So scientifically, there are a lot of models in mouse, but also in human, where simulate sepsis in healthy donor and then analyze the cytokines kinetics. And one of the key features is this blocking signature of TNF. For example, TNF is not a biomarker that you can find in any clinical laboratory because it's very unstable in blood and it's very, very early. So usually it's not useful for diagnose or for diagnostic of sepsis. So because that we create this device to simulate, the device in the end is simulating this initial sepsis because the container reactive with endotoxin to do this stimulation. And if the whole blood from the patient are able to stimulate and produce this TNF-alpha, means that their whole blood are naive or are not in contact with bacteria. So with this logic is how we create this device.
It makes sense. Okay, perfect. SeptiLoop is CE marked, I should say, under IVDR Class C and UKCA cleared. Now, which is not trivial. I mean, it's quite significant. And for particularly for an innovative assay like yours, what were the toughest regulatory or clinical validation hurdles you had to overcome?
Yeah, I would say that like the toughest one would be because we started in these times that there was like this swap from IVDD to IVDR. So the toughest was the uncertainty. As you know, like a startup like us, it's full of uncertainty. By being the notified bodies also uncertain complicates a lot let's say the milestones and the schedules that you have proposed for your business plan. So here what it happens is like we were late to regulate our product as IVDD because we didn't have like the final industrial version for that time. So we needed to enter into the IVDR. And when we entered into the IVDR, the problem was the lack of notified bodies that were certified to be able to approve technologies and to examine it under IVDR. So here our toughest pain was like the waiting time. The waiting time that so we entered in a kind of a queue for the notified bodies that were available. And this mean 1 year to 1 year and a half, almost 2 years of just waiting.
It's a waiting game, I agree. So you're positioning this as a point of care tool, which I particularly like. And what are the real world workflow implications in, say, an emergency department or an intensive care unit? And what resistance have you encountered on the frontline staff, if any at all? I don't know if you have.
Sure, it depends in the end of the healthcare system. So here, what we are doing by making our product being certified under point of care near patient testing, it's just giving to the physician and to the healthcare systems more opportunities to make to adapt this product to their workflow. So like the way that we envision this, for sure, we would be evangelists of our solution, and we will always give references in how to do best practices. But in the end, what we want is a product that is easy to use, easy to implement without any training that can be used by any type of professional. So there is like the most of the options possible, so it can be adapted to each specific case. It has been how we envision it, not just like the development of the product, but also like the regulation of the product. And here, for example, something that happens, like some restrictions, let's say, that we might find is like our test, even though that it happens very early on the development of sepsis. So it can, as you commented before, it can detect the sepsis from the first hour that the infection generates this dysregulation on the immune system.
Yes.
It needs 2.5 hours of incubation, the procedure
Yes.
of the diagnostic, because we need to mimic this sepsis outside the body, to mimic this infection outside the body, and see how the immune system reacts, let's say, again. And this time, for example, in some specific health systems for emergency department, it's too wide or too complicated, let's say, because like usually emergency department, it's a busy place, and usually there are some healthcare systems that are, let's say, lacking, or they don't have like all the personnel to acquire and to be molded with all the point of care solutions that could be made over there. So in some specific healthcare systems, what we find is it's too long of a procedure to be performed in emergency department. So usually in these healthcare systems, what even that's happening is like the product it's prescribed at the emergency department, but the protocol and the test is performed in the laboratories. So for example, this has been a solution to overcome like this restriction, let's say, that, or this constraint that we found.
Okay, so looking back on 2018, 2019, and your first proof of concept study in Barcelona, what was the pivotal moment when you both thought this is going to work? This is a company, not a project.
So for a diagnostic product, they have like 3 pillars, no? That those are the regulatory pillars: the scientific validation, the analytical validation, and the clinical validation. So in this initial pilot, we already have the scientific validation because my background, but we have to test in a prototype process this idea, no? That is possible to create a product behind this idea. So this was happening in this early pilot study in the Belviche Hospital. Then the analytical validation, we reduce this risk subcontracting the production of the expertise of the lateral flow. So also the company is moving very secure to a very robust product because this subcontract abilities. Also, and the other founder, Joan, is joined to the company at that moment to a lot with experience in product and regulatory, and they help us to also to create from this scientific idea or prototype product to in a more realistic product in the end. No? So this is more or less. And the clinical validation, from the first moment, we start doing this validation with the prototype, with the final industrial product, and now we are still doing that because there are a lot of questions pending for the product. So the product have a good performance, for example, in neutropenia oncologic patients. So now we are in this process of clinical validation, more oriented for clinical utility and to match, to see what is the real match with the specific needs of patient and is in a specific healthcare area and physicians.
Okay, that's an excellent answer. Thank you. So for me, or for a lot of people that we deal with, antibiotic stewardship is a huge theme in sepsis, as you know, and there's a lot of people in this bubble who are making a lot of noise. How do you see SeptiLoop influencing antibiotic initiation, escalation, and de-escalation strategies over the next 5 to 10 years?
So here, with this product, we want to change the mind of just to looking for bacterial detection, because when you have an infection, it's because you have a bacterial infection and because your immune system is not properly responding. So we believe that with this new tool will open the door to analyze a new dimension if the diagnostic that is also included in this detection, how is the immune system responding of the patient. So this means that, for example, with a suspected septic patient with a good immune system that is working because SeptiLoop is providing the result, maybe it's not needed to escalate the antibiotic to broad spectrum to intravenous. And the other part, if in case to the SeptiLoop identify this susceptible patient to develop sepsis, initiate this antibiotic, spectrum antibiotic, also will be work better that initiate the antibiotic where the patient are under organ damage severe. So you can eliminate better the bacteria and in an early phase before to wait. No, this is how we envision that this device could work or could work in this new trend of antibiotic resistance.
Perfect.
Also something that we are finding is like, for example, a lot of products like, let's say, blood cultures or other molecular devices that focus on pathogen detection. They are also, they have the problematic of if the patient are treated with antibiotics before. So that giving usually like less positive cases because like in the end there is not enough bacterial charge to be able to make the results positive of these solutions. And what ends up happening on what we see in most of cases is like the physicians start treating with antibiotics and escalating these antibiotics, even though they do not have a clear answer if there is a bacterial infection or even which type of bacterial infection. Like they are more looking at symptomatology and how the patient is feeling. So here we want, as Enrique well commented, we want to provide some kind of information before they do these changes.
Yes.
And like move the physicians to being used to not have a positive test before they have to guess which type of antibiotic. So like retire this default, let's say, actuation that it's not led by a diagnostic solution with a clear result.
I think using TNF alpha is a very clever idea. Okay, thank you very much. I think that for me, the thing that interests me both about all of you, the starters on the team side, one of you comes from pharmacy and entrepreneurship, and Enrique, you come from immunology. So how do you split the CEO and the COO roles in practice, and where do you meet often? Do you disagree on a productive way forward? You must have interesting conversations.
Yeah, so I think we have even the different like backgrounds. Also, we have different like way to work. So, for example, how do we manage right now in the company a new distributor? No, for example, so the scientific profile and the answer about clinical, commercial, etc. is I usually manage. Also, this answer can be answered quickly because they want to know feedback because the physician asks. So usually it's my charge in this case, a part of the organization of the company, blah blah. But this is like in this example. And in case of Eduard, is leading about the legal, the contract, with more calm spirit doing the negotiation behind this, because also it's very important because if it's to sing a proper collaboration agreement with very clear rules, etc. Also is key to have a good agreement with the distributor or other companies. No, I don't know, Edu, you want to join?
Enrique, come on.
Yeah, for sure. No, I totally agree with Enrique. And since the early beginning, it's been a little bit tricky in how to like separate the let's say the operations and the works that every one of us do, because in the end we are a very little force from the early beginning. So we've been two at the start, then three, and till now we are just getting a maximum of 7 to 8 people. Here what we've done usually, like we chat a lot. Like during the day, we always have like constant calls in most of our decisions. And in the end, Enrique is the CEO, so he has like the definitive, let's say, decision. But my job here, if I think like there is another option to reconsider or that's worth that's worth like taking the time to changing his opinion, like that's my work. So if I think it's worthy to take that risk, and if it's not the case, Enrique is like the most pragmatic person I know. So I know that he's gonna move fast. So this is usually how it works.
Okay, so I think thank you very much. I think finally for founders building deep tech diagnostics today and going down various pathways that are not necessarily compatible with patient pathways. So what is the one big lesson that you've learned over since 2018? What would be the one big lesson that you have learned?
Yeah, well, from the early beginning, like when we started the master degree with Enrique, what I see or what I expected is like if you have a solution that shows like better sensitivity, that reduces mortality, like in theory, like hypothetically reduces mortality, that people it's gonna want it and everything is gonna go smooth. And that's not the case for sure. So you need to show like a clear evidence that also reduces cost. That this is something like even though that it's a better solution, even though that there is like hypothetical mortality reduction, you also need to reduce costs in order to change work guidelines or to change work path, or even to have an option to chat with managers and the hospitals. You need at the start to show a very clear economic case with economic value.
Absolutely.
And the same goes for investors' money. Let's say if you want to raise capital, you also need a very compelling business case. And even though that there is a lot of money like moving into the impact, the truth is like impact it's nice, but impact goes very just right after there is a very compelling business case and not before.
Enrique?
So in our case, is from one of the mentors comment that it's general thought, but I think they have right that even though try to think big about the idea to be more think from more a big idea, because even the small and big idea maybe have the same effort and the same cost, same time. So this is also a good point, because initially when we jumped from the academia to this innovation program, the initial idea was like a idea to develop some tools that help but not so much. So it's incremental technology. So in terms of cost and time, even these small changes will also cost a lot in times and effort in the company to position this device in the market. So try to think in a very innovative way product, because usually the time to achieve the market will be the same for a big crazy idea, or maybe like a very complementary idea. I don't know. I think they have right.
I agree. Well, I want to thank you both very much for your time today. It's been enlightening and very, I think for me, encouraging. I think the pathway you're going down is a very interesting and unique pathway. So I love the chemistry. I love the biotechnology behind it. I think it's a very compelling idea for emergency departments and for wards. I can see the rationale, and I know they will. So thank you very much to both of you for your time today. And I hope that we can talk again in a few months, or once you get some capital behind you, and you can move forward, because that's the big game changer. So thank you, gentlemen.
Thank you very much.
For sure. Thanks to you, Stephen.